Right. Welcome to my talk. Today, I'm going to present a little bit about continuation methods for biological systems. So the plan is the following. So what I want to do is, before I get started, the plan is the following. What I want to show is a certain class of numerical methods, and I will illustrate these numerical methods for various biological examples. So I hope, since you're all sort of interested in biological systems and how they behave, that I can give you sort of some tools that you might use in your research. And, these tools are pretty broadly applicable, and I will try to sort of show that by broadly applying them to different classes of problems. But before we get started with the continuation methods, let me briefly say what I really sort of work on. I have sort of four sort of core areas in terms of mathematical techniques. These are sort of multi-scale dynamics, stochastic dynamics, non-local dynamics, and network dynamics. It's all sort of centered around the paradigm of nonlinear dynamical systems. And I will put up a few buzzwords. So maybe if you are not sort of interested into continuation methods, maybe you're interested into some of my other work. So I will put up a few buzzwords that maybe some of you might be interested in. So today, what we are mostly going to stay within is we are going to stay within, in the beginning, in multi-scale ordinary differential equations. And I will apply numerical methods to that. Then we will go to multi-scale partial differential equations. I will apply continuation methods and develop some new continuation methods for that. And then finally, we go to the sort of hardest case on stochastic numerical continuation methods, where there wasn't much research. And I will sort of basically, develop a new method completely from scratch. Okay. And today I cannot talk about networks. So if you're interested in networks, please just contact me and I will be happy to talk to you about that. All right, so let's get started. So what's the problem formulation? Suppose you have your favorite model. Suppose it's a differential equation, and it could be any sort of differential equation, an ODE, a DDE, a PDE. And let's start deterministic. Let's suppose you have your deterministic differential equation. And suppose there's also, in addition to the normal evolution in phase space, there are parameters in it. I mean, basically every biological model has that, right? I mean, you basically can't get without that. And then maybe you think, okay, the sort of plan could be, okay, I just simulate the thing, right? You put it on a computer, you do your favorite discretization in time or space-time, and then you just integrate the thing forward, and then you're happy you see an output and a time series that maybe tells you a little bit. But, the time series is just sort of, on my view, it's a little bit sort of exploratory. It doesn't really tell you how the model behaves, because if you would want to do that, you would have to simulate over lots of initial values, and you also have to simulate over the entire parameter space effectively. So it's sort of, you only get snapshots by forward integration, and that's where numerical continuation methods come in, because they sort of combine dynamical systems thinking of trying to find the interesting objects and then sort of getting them parametrically over a certain scale. So the idea is this. So what you really would want is some sort of typical goal would be a parameter versus a quantity of interest diagram that says, okay, I have a certain parameter, say P, and what I want is an observable or a quantity of interest that I can compute for this parameter. So you say, okay, maybe I want to observe, I don't know, the total temperature of a system, or a certain type of population in a system, or the total number of, say, infected people in a population in an epidemic model, and in steady state, say, and you wanted to see how that varies in parameter space. And then these diagrams are sort of, I also feel, biologically more informative, because you focus in on the dynamical quantity of interest, and then you try to compute it depending upon P. Yeah? All right, so what are deterministic numerical continuation methods? They are actually built methods how you can actually compute effectively sort of sets of zeros of a predefined problem in a sort of clever way that's related to dynamical quantities of the original underlying differential equation. So let me start with a classical problem that every sort of, I think, math biologist faces at some point. You have an ODE, you have, say, a parameter, say now a single parameter P for simplicity, and you are charged with the fact is, okay, I want to compute all the equilibria depending upon P. So let's suppose we can parameterize these equilibria by curves, because if you vary a parameter P, generically, you will vary the equilibrium location on a curve. So the task would be to compute this curve, meaning you have to compute a parameterized curve that depends upon, say, an arc length parameterization, for example. And this curve, this solves just the right-hand side equal to 0. How would you do that? So numerical continuation methods do a clever way. They do it in a sort of tricky way by saying we use a predictor-corrector scheme. So suppose you have found one equilibrium point at least for one parameter value. That's usually quite easy to guess. At least in biology, normally that's not a problem. And then you want to compute an entire curve of equilibria. So you start at one point on this curve, and what you do is you first compute the tangent vector to this curve. How do you do this? Well, for example, for this equilibrium finding problem, it's actually quite easy to see that if you differentiate this implicitly with respect to the parameterization parameter S, you see that, well, this just gives you by the chain rule this, and then you just see that this tangent vector to this curve is just a solution of a linear system. And the linear system is just basically sit at this point, you compute the Jacobian at that point, and then you solve for the tangent vector. So this can easily be computed. And once you computed the tangent vector, you make a prediction for a new parameter value. So P is again the parameter effectively you could think on our horizontal axis. You make a new prediction, and then after you make the prediction, you're off the curve. Yeah, so you're off the curve of equilibria. And what you do is then a correction step. For example, by Newton's method, you're iterating, and there are many different ways of how you could set up the Newton. But suppose you set it up in a correct way, and then you iterate back onto the curve, which is very fast, because Newton has a very good starting point that's very close to the curve already by this tangent prediction. So this is the sort of whole power. You basically run a Newton method again and again and again, but with extremely good starting data. Yeah? And that way you sort of effectively trace out the discretization of that curve. So that way, by a numerical continuation method, you can quickly compute branches of equilibrium points based upon parameters. So sorry if I bored you with this, if you know a little bit about numerical continuation, but that principle of predictor-corrector, efficient iteration scheme that's highly convergent is sort of the power of this numerical backbone of this continuation scheme. All right, how to actually sort of use this? Well, first of all, you need some software. There's plenty on the market which implements many of these things. Yeah, there's Auto, which many of you might know, and there's the sort of graphical user interface for Auto, XPBOut, which are sort of classical tools from the 1980s and 1990s. Then there's MatCont, which is a little bit newer, like the 2000s. Then there's the 2010s. There's Coco, which is an even sort of more advanced continuation package. And there's also stuff for PDEs, so PDE two-path. You could think that the same trick sort of should work with PDEs. So think about that this would be a PDE. You discretize the right-hand side, say, spatially, then you have, say, lots of ODEs, and then you just apply the same sort of trickery, and then you find sort of a pattern solution, for example, for a PDE. All right, so there's plenty of software, but you need to sort of tweak and set up the continuation problem in the right way, meaning you need to sort of get the right root finding problem that depends upon parameters. All right, so how to apply this? Let me start with an epidemic model, just to illustrate it a bit. I mean, it might be quite timely to look at epidemic models, yeah? So this one we actually did already in 2019, and we published a paper in 2019 that got accepted after a while of reviewing, actually, where we looked at the waning immunity SIR model, where you have also a state of waning immunity, meaning you have a normal SIR model plus a waning state, and after the waning state is over, you go back to the S compartment. If you write out the sort of simplest mean field equations, assuming sufficient mixing, yeah, we also did this on a network, but I'm just presenting the homogeneous result here. Suppose like basically everybody talks to everybody, then you get a closed ODE system. And let's suppose we look at the dependence in this ODE system on this parameter P that sort of controls how you go from the recovered to the waning compartment. Yeah. So you can basically vary that parameter and ask, okay, what sort of behaviors could this model have if I vary P? You know, normal SIR is, reasonably sort of simple. Normal SIS is also reasonably simple. But what's the parameter space look like if I start to vary this P? Yeah. And we also did more. We proved also stuff like by pencil and paper, but let's just look at the numerical continuation output. Here's a typical output, what a numerical continuation algorithm would give you. You see this parameter P on the horizontal, and the sort of black curve is a curve of equilibrium points. you follow and track this curve of equilibrium points, the algorithm is also able to tell you when there's a bifurcation point. Why? Because, well, bifurcation conditions you can actually write as zero conditions, basically. And since you can sort of try to detect zeros of certain conditions with continuation algorithms, you can also, when you are clever enough in some sense, detect certain bifurcation points. And here it detected two Hopf bifurcation points where the equilibrium loses stability. There's a subcritical Hopf, a supercritical Hopf, and you see that there's actually regions where there are limit cycles. Yeah? There's actually here, there's even more. There's sort of a limit point of cycles, yeah, where the limit cycles disappear again. But yeah, but it gives you a nice view, and it says, okay, for certain quite broad ranges of P, there is actually a limit cycle, a stable limit cycle that seems to oscillate. Yeah? And indeed, you know, you could prove that. We could prove that over wide ranges of parameter sort of choices in this model, you have certain types of limit cycles. But the numerical algorithm scanned you the parameter space and told you where you should sort of look. Yeah. And that's the great thing about numerical methods. You can easily check quite quickly that says, okay, look, where do I have something interesting? And it gives it to you. Yeah? And here, the interesting part in that model is that it starts to oscillate. Yeah? Also not so surprising, because somehow you could think that there might be sort of a balance where you are very close to nobody being infected, and then you're sort of slowly waning with the immunity. And at some point, right, there is just enough accumulation of initial infected, but then very drastically and rapidly, you somehow again are in the phase of that a new epidemic can start, and then you have a big sort of cycle, and then you do this again and again. And these are, again, these cycles that you see in these waning immunity models. We did this pre-COVID, yeah? So, and seems to be that, you know, might not be that useless after all. Yeah? Very good. So here's a more complicated example now from neuroscience, just to show you it's not really depending upon the biological application. So this model is the sort of three-dimensional FitzHugh-Nagumo equation, which comes out of the actual PDE FitzHugh-Nagumo equation that I've written in the traveling wave frame. So recall FitzHugh-Nagumo models the propagation of action potentials in a simplistic way in axons. If you write this equation in a traveling wave frame by a spatial dynamics ansatz, it basically becomes ODEs. And then out of the two PDEs in the first coordinate, you basically get two ODEs. And then you could also ask the question, okay, for these two ODEs, what would be the, for these three ODEs, what would be the bifurcation diagram? Yeah? Could you compute that? And indeed, you can compute that. So this is in these papers. I computed that actually in my dissertation. I just wanted to show this is sort of good old fun. You can also compute bifurcation diagrams with two parameters. Yeah. So for example, here you see in green a curve of Hopf bifurcations. Before we had a Hopf point. If we make one more parameter free, we get a curve of Hopf bifurcation. You can detect a codimension 2 bifurcation, a generalized Hopf or Bautin bifurcation, yeah, where you have an emergence of a saddle node of limit cycles. You can detect the homoclinic bifurcations, all this sort of stuff, the numerical algorithms, after you set it up correctly. You can write all these problems that these curves are given here as zero finding problems. You can continue all these curves in parameter space, and you can trace out the picture. For example, here it would say, here there's like little excitable sort of traveling waves that are sort of wave trains bifurcating. And if you move from the wave trains, eventually the wave trains, if you move in parameter space, they will deform into actually a pulse. And here in the vicinity of this homoclinic, you effectively see on a long time scale for these parameter values P1 and P2, you see pulses. And even in this homoclinic curve, you can even resolve what types of pulses you see. Yeah, that's very nice. I mean, I haven't plotted the pulses here because then the whole figure would be full of all sorts of objects. But you see that that's sort of good fun. Yeah? All right, so you can do one parameter, two parameter, and all sorts of stuff with ODEs. But let's be a bit more ambitious here. Let's do PDEs instead. Yeah. So again, I wanted to illustrate these methods are broadly applicable. So I thought, well, we have done epidemics, we have done neuroscience, and you can also do this for ecology and population dynamics. Again, take your favorite model here. I took my favorite one, the Shigesada-Kawasaki-Teramoto, the SKT model, which is a sort of competition model of two species, which has a speciality. So you have two species that are competing for a resource. But there's also sort of diffusion, and the diffusion is sort of in some sense non-linear. So there are terms in there that are not standard diffusion, and they're density dependent, meaning depending on how many people in this sort of species or how many animals in this species you have next to you, the diffusion depends upon the local density. So this sort of models crowding effects, for example, that the way you diffuse is not completely independent about the concentration of other people in your environment. Yeah? And this is a well-known classical model from math biology. And we were interested in, okay, how do the patterns actually look? Yeah, what sorts of types of solutions can you get? In particular, can you get solutions that somehow show crowding or spikes of people or animals competing for that resource? And indeed, you can get them. So each of these sort of branches that I plot here, depending upon P1, I just plot the middle value in the domain of the second species. Yeah, I could plot the L2 norm, I could plot all sorts of other stuff, but you see that there's a sort of stable branch here that's solid, that starts to bifurcate, and then there's a new branch again that's stable. This initial branch becomes unstable, and then on these sort of non-trivial branches, all these solutions have different pattern structures. Again, I haven't plotted them all here, but if you think about them in space, they're sort of in space, they're very sort of spiky and patterny. So you see that from this sort of diagram, you can sort of read off what sort of patterns varying when you vary this diffusion parameter P1, you can actually get. Yeah? So of course, you would argue, okay, locally, these first few points, that the bifurcation from the homogeneous state, this I can get from standard Turing analysis that everybody learns in math biology, I don't know, in lecture 4 or 5 or whatever. Yeah, that's true. But these global curves, like how they bifurcate and how they deform globally away from the first points that are the local bifurcations, this is by no means obvious how to compute that. Yeah? All right, so you can understand the influence of cross diffusion. And well, these bifurcation diagrams in PDEs, particularly, they can get quite complicated. So I want to show a few more just to give you an idea. For example, here, by the way, this is unfortunately an error. This typo I just noticed before I started. So this should be the L2 norm of the first component. Yeah. So this is the L2 norm of the first component, and I plotted a parameter again P1, and I vary also P2, which is the second parameter here. And then I look at how these first branches that bifurcate off at the first sort of bifurcation point, how they vary if I vary the second parameter. So there are multiple bifurcation branches now plotted, and you see, well, you can get regions of sort of multi-stability, you can get branches that sort of topple over, that where the multi-stability is gone. You can get all sorts of stuff depending on, you know, these diagrams, and you scan the parameter space, and it exactly tells you where different types of pattern solution exist. Well, even better than that, you can also like understand analytically certain things in some sense better and why they work. So here's a 3-dimensional sort of variant of the SKT, where there's sort of a different type of that the first sort of species that actually is now allowed to have one species that's a little bit sort of more active than the other. So this guy, the second guy, has a little bit more diffusive action, so it's a little bit more running around than the first one. And the question again is, okay, what are the patterns? Well, theory tells you that if you send epsilon to 0 in a so-called fast reaction limit, then there's sort of proofs that tell you, okay, somehow this first big system of 3 should be approximated by this sort of smaller system of 2 equations. Yeah, some sort of limiting SKT model should come out if we here look at the multi-scale nature of this sort of fast reaction, meaning the first guy having a very fast reacting in comparison to the second. Well, we were interested in, okay, how do you actually, in this sort of model problem, compare, for example, the stationary solutions of this complicated system with the stationary solutions of this simpler system? Well, you could try to start doing hand calculations, but of course that's very complicated because, you know, even, I mean, for the homogeneous branches, you can do it, but once it becomes non-homogeneous, and then you also have to take into account how epsilon is varying, it would be very difficult to compare all these patterns. Well, again, you could do continuation. So you discretize basically the right-hand side, you write it as a 0 finding problem, and then you solve it. And then you see, okay, now we have this bifurcation diagram for epsilon equal to 0.05 of the 3-component model. And then we vary epsilon, yeah, 0.01, 0.0, 0.001, and then 0. You see very nicely that this diagram now is understood, meaning, well, even if epsilon is quite small, there's quite a difference in the bifurcation diagram between the two models, at least sort of quantitatively. Yeah, so you need to go down quite a bit, at least to 0.01, to see the resemblance really of the diagram that we had before. And only if you go really to 10 to the minus 3, effectively, you see that somehow you are very close to the limit. Yeah, so these last two are not very distinguishable. But even there are sort of little things that change, yeah? Particularly if you go with the parameters also to a singular limit. So this is nice because it also tells you something that the theory cannot tell you, namely how big the epsilon has to be so that you in practice really, at least for the steady state, see something similar, right? Theory will tell you there exists an epsilon naught, but for all epsilon sufficiently less than this epsilon naught, then this sort of limit exists. But that in biological problems might not mean very much, because you might want to know how big this epsilon actually is. And the numerics gives you an efficient tool to get that out. Yeah? All right, so you can do even more. Yeah, you can also find new features. For example, now this is the first branch, a little bit continued upwards away from the homogeneous state. So we are now away from any sort of Turing analysis, effectively, really. So you can do that, and you vary the bifurcation diagram with epsilon. And you see if you make epsilon smaller, really sort of funny structures seem to emerge, sort of new solutions that are really looking. And then there's even new bifurcation points emerging. Yeah, and even more, and then these are deforming. And if epsilon is even smaller, at some point they're reconnecting again, and then they're sort of breaking up. Yeah, I always like to call this sort of the broken heart bifurcation in some sense. So you see that even for tiny epsilons, theory would tell you everything is sort of at some epsilon close, but the epsilon is actually, you see this here. It could be that you need in sort of, to make this theoretical result applicable, you might need something like 10 to the minus 10 or so. Yeah, but in practical terms, lots of stuff happens when you move towards this epsilon equal to 0 limit in this model. And numerics can tell you this quite easily, namely that all these patterns are branching off from the initial branch. Okay. So you see that, let me check my time very quickly. So that was sort of a little bit of an introduction to numerical continuation methods, and that you can use them really very effectively in biological models to find stuff that theory would never tell you. And also forward simulation would never tell you, because these steady states of patterns, if you wanted to find them all with forward simulation, it would take you forever. Yeah? And this runs on a standard laptop in like 10 minutes. Yeah? So there's really a big gain here. Very good. So now we come to the sort of hard part. Now, if I bored you till now with saying, oh, yeah, I could have done that myself. And yes, you needed to be a little bit more clever for the cross diffusion thing. And yes, you needed to discretize that a bit more cleverly. And yeah. Now we come to something that sort of needs actually a little bit more brain power than just trying to generalize existing algorithms. So in the second part of my talk, roughly the second part, so we will also be roughly 25 minutes, I want to explain you that sometimes you have to rethink continuation from the ground up, that there's no algorithm yet available and you need to design one. And this is a story about how to extend all of this to stochastic systems that I've shown you so far. You have seen that for deterministic systems, this is well developed, but for stochastic systems, there's actually not much known. And a while ago, 5, 6, 7 years ago, I thought, okay, we really need also something for stochastic systems, in particular for biological models, right? Because you know that somehow, the models I've shown you really in practice, there should be also stochasticity. But then it's even harder. Why is it even harder? Well, you not only have to simulate over different initial conditions, you not only have to simulate over parameter space, but you also have to sample different noise realizations. So if the right-hand side would be noisy, then it would mean either, the parameters would be even more random, or there would be stochastic forcing, which is the case I'm interested in. You would need to sample also over all sorts of different realizations. And then you have to average all of this. And, it's all very, how to say, non-fun, because do you really then understand your system very well if you do that? And I think we should be able to do better. Well, again, the goal of this is parameter versus quantity of interest. Also for a stochastic system, you could plot an observable. The average number of infected, the the variance of some quantity, like, I don't know, the predation of a certain predator in a certain part of the environment, for example. So you could measure all these things, and you again want the same sort of diagram. Parameter value versus something that you actually can biologically interpret. But now we have this nasty point 3, that there's all this noisy stuff. How to do that? Well, let's start with stochastic ordinary differential equations to illustrate the problem. Yeah, so let's take an SDE. We have our normal ODE here plus some stochastic forcing. I will assume that this will be, for a simplicity, say, IID Brownian motions, and then some potentially multiplicative noise. And I will define D. This will be a sort of standard diffusion tensor or diffusion matrix. And then the question is, okay, what can you do with these guys? Well, the first thing you might think, I do it by sampling. Yeah. So I do it by sampling, and I will just do the same thing I did for ODEs. That it sort of worked for ODEs sometimes, and I can do it by sampling. But sampling is now so prohibitive that the only method that people tend to use is this, what I have called the GSMC method. Can people think what this GSMC could mean? Are there any guesses from the audience what that could mean? What could be a GSMC method? Half of that you could guess.
Like a generalized Monte Carlo?
Pretty good. It is, yeah, this is already, this would give definitely an A, maybe not an A+, but it's already an A in an answer. It's a generalized type of Monte Carlo method. But what I called this because there was nothing. So what you normally do is you look and you say, well, I need someone to actually do all these simulations, and it takes forever to do this. So how do I do that? Well, normally the people who do this are the graduate students. So I thought, okay, let's call this the graduate student Monte Carlo method. So you sit somebody who just sort of ticks the boxes and then simulates over different parameter values. And I found this not very efficient. Yeah. So I thought there must be something better than just simulating over everything and having somebody plot this. Yeah, this is not very mathematically efficient. So then you could think, well, maybe I can use a probability density. I could say, okay, maybe from basic course on stochastics that you can, instead of tracking just every path, you could try to get a probability density function for the stochastic process and solve a Fokker-Planck equation. But that Fokker-Planck equation is a PDE, and that PDE has the same spatial dimension as the dimension of the original equation, meaning that, for example, if X would be, say, 5-dimensional, then this would be a 5-dimensional PDE in space. Yeah, that's already almost hopeless. If you have even more, like a really big complex biological system, this might be a 100-dimensional PDE that might be highly nonlinear, and that's basically intractable. Yeah. So doing the PDE is somehow out. But before, what we are focusing on is not trying to solve and simulate the entire system and get the entire sort of description. But what you wanted to do is you wanted to just understand certain types of the dynamics. And that's what I'm going to try to tell you. There is a way to do numerical continuation for these sort of stochastic problems if you're only interested in part of the dynamics. And you can do it efficiently on your laptop, actually. So how to do that? Let's suppose we are only interested, in the behavior near locally deterministically stable equilibrium points. So suppose, for example, we have already computed a curve of equilibrium points, and we only would be interested in, if I added on these fluctuations, how big would the fluctuations be, and in what directions the fluctuations would be acting. This makes sense because in a biological system, you would think, okay, I have some fluctuations around in equilibrium, and I'm interested how big they are and what are the most important local perturbation directions, how I can deviate from what I have in the deterministic case. So let us assume noise is sufficiently small. If the noise is big, you probably didn't model the system right. So let us assume that we are just interested in sort of small noise, but we're interested in what directions are sort of relevant and what directions are not relevant. Yeah? So what do? Well, we first take the deterministically stable equilibrium and we tailor. Yeah? After we tailored, we have basically a local linearization of the stochastic fluctuations around this equilibrium point. Yeah? Okay, this gives you this stochastic differential equation. So here I just tailored, this is just the Jacobian, and here I just evaluated the leading order term of the noise. I suppose this is non-degenerate. I mean, generically, it's non-degenerate. So, and then this sort of process is a well-known so-called Ornstein-Uhlenbeck process. And you can also figure out that the covariance, so the fluctuations of this Ornstein-Uhlenbeck process, are completely described by a covariance matrix. Yeah, so Gaussian process, mean zero, and so on. You can actually find the covariance matrix, and the covariance matrix solves an evolution equation. And that evolution equation for a locally stable deterministic equilibrium can be proven to have a stationary solution. And this stationary solution solves this equation. AC plus CA plus some other matrix is equal to 0. So this is basically the long-term local stochastic fluctuations around the equilibrium point. So if you could compute this matrix C that solves this second equation, then you would be done. Because roughly speaking, if you take this matrix C, you could define in space what is called a covariance ellipsoid, or statisticians would call that the Mahalanobis distance. Yeah. So this is basically a shape around your equilibrium point that tells you in one direction there's lots of fluctuations, and in another direction there might be not many fluctuations. So if you could compute C, you would have won. I will show an example later on. Yeah, but if you could compute C, you would have won. So how do you compute C? First of all, well, you compute your normal deterministic equilibrium branch with continuation. Pretty easy. We have done that before. But for the covariance ellipsoid, you have to solve an equation for C that has this form. But this is actually sort of standard equation in some sense in linear algebra or matrix algebra. It's called the Lyapunov equation, or sometimes the Lyapunov-Riccati equation. And this equation you have to solve. But that equation, well, you could solve again by numerical continuation. How? First of all, these matrices A, they have been built from the Jacobian, and B is just an evaluation of the noise. So this matrix here, A, that comes from the Jacobian, you have already built in your first step of your continuation algorithm because you computed these tangent vectors along the branch of equilibrium. So therefore, okay, this A is available, B is easy to evaluate because you just get this by direct evaluation of the noise term. And then you have all these matrices, but even better than that, it's known that they're efficient iterative methods for Lyapunov equations. And a simple initial guess for your matrix that you are trying to find is just the matrix from your previous sort of parameter value. So you can do basically predictor corrector like you did before. You can tune that, there are better and more efficient initial guesses and so on. Yeah, fine. But you could just say, okay, in parameter space, I take my covariance matrix that I have for one parameter set, I move my parameter a little bit, I compute my new equilibrium, and then I compute by an efficient iterative method my new matrix. Again, the sort of newly iterated matrix that I got after sort of my predictor step. And that way I can trace out all these matrices despite the fact that they are large dimensional, so squared dimension of your system. I can compute them extremely quickly because I've harnessed the power of continuation and I've rethought how you could do a continuation algorithm in the stochastic case. Yeah? So this is what I mean with rethinking continuation from the ground up. You sort of reset up a new problem, you figure out how that fits into this continuation predictor corrector iteration scheme, and then you efficiently can solve it numerically. Great. By the way, you can also compute distances between ellipsoids. This is for the experts in numerics. I will only briefly touch upon that. So this is, in some sense, you can rewrite ellipsoids also in this sort of different form. Yeah, so if you have a positive semi-definite matrix that defines an ellipsoid, you can rewrite that ellipsoid in this sort of form via something called ellipsoidal calculus. And if you do that, there's a fact that tells you if you want to compute the distance between two ellipsoids, you can do that by solving an optimization problem. But now if you have the problem of finding sort of an optimization problem, again, you can do that by continuation. Suppose you have solved for one parameter set already the distance between two ellipsoids, you could go to the next parameter set and start your optimization problem precisely at the initial guess from the previous one. And then you can use an iterative method like, sequential quadratic programming or so to solve your optimization problem. So this is rather cute. You see that somehow every sort of, problem, you can just sort of rethink it properly in this continuation framework. So let me do an example. Yeah, so I've now sort of bothered the biological part of the audience with all this numerical detail. So let's take a simple model of binocular rivalry. Remember, this is when you cover one eye and then you cover the other eye. What do you see? Well, Yeah, you don't not only see sort of one and then the other, you see sort of a superposition. So there are two competing neural populations, and for simplicity, let us just assume that these sort of slowly varying variables y don't play a role for now, and you set this epsilon equal to 0. You just have these two neural population with sort of that are competing, and there's one parameter that you are varying. Yeah, there's a typical, you know, firing function and everything that you know from neuroscience, that's all there. And you're just trying to interested in these sort of neural mass models of two competing neural populations. And look at the subsystem, as I've said, and let's add on some noise. Yeah, I haven't modeled this in detail, but you could model that, of course, by system size expansion from microscopically deriving that these neural populations would be noisy. I'm just doing this to test my numerical scheme. Okay. So what you find is the following. So here's the bifurcation diagram that illustrates the method a bit. So this thick red curve is a deterministically stable branch of equilibria. The same is true for this blue curve. It's a locally stable deterministic branch of equilibria. The green is an unstable branch. There are two fold bifurcations terminating these sort of this blue branch. You see here around this red branch, around this blue branch, I've computed for a certain fixed additive noise. I have computed these sort of covariance ellipsoids, which tell you somehow in what directions the perturbations are most likely to act. For example, here you see that the ellipsoids actually deform, and sort of this ellipsoid is deforming in a certain direction. You see, okay, if I'm really close to this fold bifurcation, probably most likely I'm going to get kicked in this sort of downward direction here, and not really in this sort of more horizontal direction here. And you can also, here is a projection on the top right in the parameter space, and only I've looked at the x1 coordinate. You again see the two locally stable branches. I've also projected the ellipsoids, and I've also computed the distance between these ellipsoids. So you can also say, well, if these are overlapping, this means, you know, the small scale fluctuations already bring you quite easily from one branch to the other branch. Yeah, because already with the local linear approximation, you are very likely to go from this branch to that branch if these are overlapping. And this distance has been computed by adding on the optimization problem and the sequential quadratic programming via a predictor corrector iterative scheme onto the normal continuation method. It's pretty powerful. You just analyze your stochastic system basically in like 10 seconds on your laptop, and you get a plot like this. Yeah, so this is pretty good. I mean, if you wanted to prove that by hand, good luck. And if you wanted to do that by forward simulation, well, graduate student Monte Carlo might take you 6 months until the student comes back and produces that figure. Yeah? So also not the most efficient way of doing it, I think. Very good. So you see how powerful that is, but you can even push this further. You can do this now also for stochastic PDEs. Yeah? Let me check how much time I have. Yeah, I guess I can do about 5, 6, or 7 minutes on stochastic PDEs. I guess that should be fine. So you can do that. This is just a toy problem, the Allen-Kahn problem. You can reinterpret that as a sort of Nagumo equation with a 5th order term if you are in biology. Yeah. And the Nagumo equation not on the line, but the Nagumo equation in 2 space. Yeah. So this is in a spatial domain, say on a rectangle, for example. Yeah. And again, suppose you wanted to do this for an SPDE. How do you do this for an SPDE when you wanted to compute with a sort of stochastic continuation method? First, you compute the bifurcation diagram for the PDE. Yeah, remember first the skeleton here was this sort of this thick red curve, this thick blue curve, and this green curve. This was deterministically computed. Yeah, you can do the same thing for the PDE. You just need a more complicated scheme, but we have already seen in an even more complicated PDE model for the stationary states for the cross diffusion model in the sort of, well, middle part of the talk that this is possible. Yeah, and I've used PDE 2-path here for that. Yeah. Afterwards, you consider the SPDE version. So you model the SPDE, you add on the noise that you like. Yeah. Derive it, for example, from first principles, derive it from system size expansion, derive it from external input, whatever you want. Then you discretize in space. So if you discretize this guy in space by finite differences, finite elements, spectral Galerkin, you get a huge system of stochastic ordinary differential equations. Okay, you look at this huge system of stochastic ordinary differential equations. And, you know, after you did that, then, well, you apply the numerical continuation tools I've shown you before for SDEs, for stochastic, or SODs, for stochastic ordinary differential equations. Yeah. Okay, let me try to do that. So here's the output. First, that's the deterministic bifurcation diagram for the Allen-Kahn equation. So you see here a stable branch, it subcritically bifurcates. There's a fold, it restabilizes, there's a region of multi-stability. So this on this branch, actually on B, you get these double bumps. Yeah, here the sort of, or well, on B, here B, sorry, you get a single bump. In the second bifurcation curve here, you get the double bumps and so on. And you can continue these branches deterministically. But now let us suppose we are having a single bump solution B and we are decreasing our parameter P downwards. Eventually, we will fall down here and the pattern solution, for example, think about this as a vegetation pattern, for example, in a more complicated model, would disappear if I decrease my parameter, and at some point I would fall off and I crash down onto the desert state. Yeah? How would you actually be able, for example, to understand how the stochastic fluctuations behave near this fold bifurcation? Well, you could immediately compute that by my stochastic continuation algorithm that I've developed. So here's the answer. So this is varying P downwards, and this is a suitable norm. Yeah, basically, for example, an L2 norm, and around the L2 norm, a suitable spectral norm of the covariance operator. And you see here, the closer and closer I come to this fold bifurcation point, which is this fold bifurcation point here, the bigger and bigger the different types of stochastic fluctuations get. I've plotted here the stochastic fluctuations for different noise sizes. So you see that the closer and closer I go, there's more and more fluctuations the closer and closer I get to the bifurcation point. This is actually sort of theoretically known. There's a theoretical scaling law that tells you how these fluctuations should behave. And you could also use that as an early warning sign to detect that point, even for an SPDE. So here's a little bit of a proof of concept that my algorithm computed that correctly. So this sort of thick black line is actually a scaling law with a factor of -0.5 in the exponent. The dashed line is a scaling factor of -1. These are the two most common scaling laws of the covariance operator near an SPDE sort of bifurcation point with sufficiently small noise. And you see indeed by theory that it should be a -0.5. In my algorithm, these are the thin blue lines, correctly computes that the exponent in this log-log plot is -0.5. So without even knowing all this theory about early warning signs and spatial stochastic SPDE fluctuations, you could have easily figured out via this algorithm how actually in this bifurcation diagram, the local stochastic fluctuations near this important bifurcation point, think about this as desertification if you wish, yeah, how this point behaves. And this computation took a couple of minutes on a standard desktop. Yeah? So you see here that these methods for any sort of biological model can be pretty powerful. And that's what I wanted to sort of advocate. But for more complicated models, like for the stochastic models, you need to rethink them from the ground up. Yeah. So here's a little bit how we rethought them for the stochastic problem from the ground up. I started to do this for the deterministic case for the stochastic case for stochastic ordinary differential equations. Then I extended this also to SPDEs in the second paper. We also applied this to climate science. I haven't shown you this, yeah, but of course it's also an interesting problem. At least, if you're math biology, maybe you're also interested in sort of broader sustainability issues. So maybe people want to have a look. And then we did a full error analysis by sort of hard numerical error analysis estimates. And we also were able sometimes to sort of link these by actually using even the computer to use these methods to construct rigorous proofs. Here we only did this for stochastic ODEs, but sometimes when you have a numerical output, you can actually use that to computer validate the numerical output. Yeah, so this is a step further that I don't have time for, and maybe this audience isn't much interested in because it's a math bio seminar. Maybe you're not interested in automated computer proofs. Probably not. There are other audiences that are interested in that. But you see here that I've sort of rethought the continuation framework for this new sort of class of equations from the ground up. And that's sort of the fun in deriving new continuation schemes. Sometimes you have to go back to basics and rethink the entire algorithmic framework. All right. For all the preprints and all the material, everything is freely available from my website that tries to be easy to remember by its name. And also, of course, from the archive. And I would like to thank you very much for your attention.
Thank you, Christian. Very nice talk.